Welcome to Types of Microscopy. This is your host, Dr. V. We're going to go through and talk about the different types of microscopy that you might find in a microbiology lab or in a university setting. It's important to know what's available to you. Many hospitals do, in fact, have higher types of microscopes that we're going to talk about. So I think it's well worth your time. When we use the microscope, of course, we have to be concerned with what we are able to do in terms of how small we can go. And we are living in the nano age down to the billionth. So that's 10 to the minus 9th, nano 9, Latin for 9. So the dimensions that we can do now, believe it or not, is really close to the atomic scale. And that's what we're after. So that's important to us. So various sizes that we need to be concerned about are usually in the millimeters or in the micrometers. Protozoa and algae are pretty large as far as micro is concerned. The smallest bacteria measure around 200 nanometers, which is pretty small, and the largest around 750 micrometers. Most viruses measure between 20 nanometers, I call them the nanometers, 400 nanometers, and some can be as big as 800 or 1500, but we still have to use a scope other than a light microscope to be able to see those. So that's kind of what we're dealing with here. So really what areas that we concerned is in this region here. Really at the 1 micron to the 200 nanometer, sort of in that range is the smallest we could do with a light microscope. And then we get into the range of electron microscope, which we'll go over. The light microscope, various aspects that we went over in the lab, but for our purposes here, we have a binocular, which has 2 eyepieces, the binocular body. We have the arm, which we carry with one hand holding the arm, and then underneath the base. We don't drop it. We have the focusing nose piece, and sometimes it's referred to as a revolving nose piece, and it has usually 3 or 4 objectives. Ours will have 4 objectives. The stage, and then we have the stage manipulators. This doesn't show that. Clips, and to hold the slide on the stage. And then things underneath the stage, we find, of course, the coarse adjust and the fine adjust knobs. The power is usually down. Ours is a rheostat, so it rolls like a sort of like a rolling switch. And then below we have features of condensers and iris and diagrams and things. The iris diaphragm is pretty much like the old time cameras, where you get the f-stop by changing the iris size and those sorts of things. Usually we don't have to mess with too much. The only thing I would check with a condenser control knob is making sure that the condenser diaphragm, the whole setup is below about 3/4 of an inch underneath the stage. That's usually a pretty good time, this size there. So, and then I think we're in good shape as far as those. Please learn the names of the various pieces and parts. And here's just another diagram showing the same thing, and you can spend some time looking at it. But the names are the same, except you have the manipulators for the stage. They call it the stage adjustment. This is how we move the stage. We have the small adjustment and the larger adjustment. Stage will move. There's the clips that hold the glass slide, and it even has some markings here that you can remember where your settings were when you were working with a slide. So if you had to leave, you could pick up and you record those positions, and you can set your scope right back the way you had it. Now, keeping in mind, of course, the magnification, you can read the magnification right off of the objective lens, and this one happens to be the 40x. The ocular objectives that we have is 10x, so the overall final focus would be, in this case, 400x. That's your final or total magnification. And now we need to report that because not all scopes are configured the same way. These may have 15x for the oculars, perhaps. Ours are 10, but there are some that do others, and then maybe the magnifications are different as a result of that with the objectives. So it's good to report the final magnification, and that way anyone that wants to reproduce your work will have what they need to achieve with their scope. Okay, so preparing the specimens, usually we'll talk about doing gram stains or other types of simple staining. We typically don't do much in the micro lab for wet mounts and hanging drops, although you could do it to look at what might be in water and that sort of thing, but we don't particularly worry about that. The staining, we use various dyes. Now, overall, a stain can be also called a vital stain or a vital dye, and these are the ones that actually bind to the bacteria or whatever microorganism that you happen to be staining. Staining is a procedure, and for various different types, and you follow outlines that people have actually worked out that work well. Basic dyes have a positive charge. Acidic dyes will have a negative charge, and that affects what sorts of things, of course, these dyes will stain to or stick to. And so we usually refer to these or the basic dyes or positive types of charge. Usually we can see polysaccharides, and acidic dyes are usually what we use for the vital stains that are repelled by the cells. And so anyhow, that's pretty straightforward. Positive stain dye sticks to the specimen, gives it color. Negative stain does not stick to the specimen but settles some distance from it. So if you're looking at, let's say, a polysaccharide around a bacteria like a pneumococcus, then you would see this silhouette or this sort of area around it. Now we have those slides in the lab so you can see those, but I just want to make it available to you. So a negative stain doesn't stick, a positive stain does. We can use nigrosin or India ink for negative type stains. Simple stains only require a simple dye. This could be Congo red, could be various different types of simple stains. The counter stain that we use in the gram stain could also be used, the various dyes associated with that particular stain. Differential stains like the gram stain use two different types, the primary and then the counter stain. And usually we want to differentiate because one binds to the other and doesn't to another type. And so these sorts of things are useful, and we try to use those when we can to help us in our diagnosis of a particular microorganism to identify it. And we'll do this in the lab many times. So here's some simple stains. Here's crystal violet used for E. coli, methylene blue. Nothing substantial, but it does add some color and some depth to the microorganism. Normally they would be colorless, and contrast would be really poor. Now the gram stain I've mentioned was developed in 1884. He had no clue what he was actually doing with the stain other than he just tried all sorts of stains. And so we use the gram stain today, and it's a sequential of crystal violet, which binds to a pretty thick layer in gram positives. We didn't know, or he didn't know, we know now, but the crystal violet will bind. The gram's iodine is a mordant. That means that it holds the crystal violet in for the gram positives. The gram negatives, there's nothing really for the crystal violet to bind to. So the crystal violet goes inside the structure of that teichoic acids that form in the outer wall, and the gram's iodine sort of blocks it so it doesn't leave when we do the alcohol rinse or the decolorizer. And sometimes it's referred to as the acid alcohol, but the alcohol just tries to get rid of the crystal violet if you can. So in the gram negatives, it does the function for that. Saffron is the counter stain, which is a simple stain I mentioned before, but saffron is sort of pinkish in color, where the crystal violet is violet or dark purple. And so if the bacteria retains the violet, the crystal violet, then we know it's a gram positive. And if we use the alcohol and destains the crystal violet, and we now counterstain with the saffron with a gram negative, it will have the reddish color. The saffron still binds the gram-positive, but you don't see it because of the masking of the dark color of the violet. It's a pretty clever stain, but it is a differential stain, and I just wanted to make sure that we got that. So the differences in the cell wall structures are helpful. If we know if the gram-positive, gram-negative, then we can make a presumption to go after a particular antibiotic, let's say, as a preemptive strike while we're trying to identify. So we'll use a broad spectrum type of antibiotic. But you can see big differences here between the gram-positives, which are here, and the gram-negative. We have a phospholipid bilayer that we see in the gram-negative. The gram-positive has this peptidoglycan with teichoic acids interspersed, and that's where the crystal violet stains. And so this, as a result of this, will stain with the counter stain, and we'll have the pinkish color, where the gram-positives, the crystal violet will stick to this peptidoglycan sheet and get caught in there with the iodine as a mordant, and then that's how we see a gram-positive. So that's pretty straightforward. And you can see that illustration here, the cell wall, where we have a very thin peptidoglycan in the gram-negatives, but it's the phospholipid bilayer that really does the trick as far as not binding to the crystal violet. So, and you can see a very staunch difference. There's a gram-positive, gram-negative, and I'll tell you, Gram did a great job with that. So differential stains, like the gram stain, there are others that we use. In particular, we use the acid fast. Sometimes the gram stain doesn't really work well, and so we want to go after using the counter stain or the carbofusion with the acid fast type of staining, and I'll show you how that works here in a second. But they have a different type of cell wall that will hold tenaciously to that carbofusion and will stick still after an alcohol decolorizer. And so this is what we see. The acid fast negative, the counter stain from the acid fast stain predominates, which is that purplish, and the acid fast positive, which is the carbofusion, that's red, and you can see the acid fast stain here would be. So it's sort of opposite of the way the gram stain is. So we're actually looking for the pink for a positive for the acid fast, and that's what we're using usually for detecting tuberculosis or something like that. Other stains that we can use that are differential are the endospore. You can see that we see various forms that are coloring, and these bigger, larger areas usually represent a spore. The typical dyes that I see most frequently is that you'll see the clearing zone at one end very clearly a spore. This is the Clostridia type of bacteria, and spore formers are highly resistant, and that's why they're problematic. The old mummy's tombs, and it's the revenge of, no, it's not. It's spores usually in the wrappings, and you breathe them in, and you can get into trouble with various things that Clostridia species are known for. So the differential stains, again, help us to distinguish between gram-positives or gram-negatives, or the acid fast if it's a tuberculosis, or endospores. And so we have a nice collection of those differential type stains. We can also use just a straightforward differential stain, the gram stain, and out of blood. So we could just do a blood smear on the slide, do the gram staining, the cells will stick, and unfortunately this patient has Staph aureus in the blood, which is not a normal condition, and that's not a good thing. We do have capsular staining, as I mentioned, the negative type staining with India ink, or using the flagellar stain, which is these little small spinning type filaments bacteria use for locomotion. We'll talk about that in class. They spin in one way or another, provided that they want to move forward or kind of tumble and move in other directions, but the flagella is responsible for that, and we have stains for those type of things, and you can see those here. The India ink has that clearing zone around the capsules, so we know Cryptococcus neoformans has that polysaccharide, and Proteus vulgaris, which is really motile, it'll swarm a plate, but you can see why, all these flagella all the way around the peritrichous type of arrangement. So on the regular light microscope, as I mentioned, we have the power of the objective and the ocular. You multiply those, there's your total magnification of 100x. And so what we're doing is amplifying sort of a virtual image in these two stages of lenses, and we, as a result, we win with the 400x or 1000x. If we add the oil to the oil immersion, we get the 1250x, which I'll talk about here in a little bit. So what I mean by the real image, of course, is what's on your slide. Then that is sort of portrayed or projected through a lens system, and the virtual image actually comes to your eye because it's been magnified by the lens system. So that's what they mean by that sort of arrangement. The resolution, or the resolving power, is the ability to distinguish between 2 points or that are distinct from one another. And so the resolving power of the human eye is about 0.2 millimeters. The resolving power of a light microscope using the oil immersion lens is about 0.2 micron, which is a significant improvement, about 10 to the third better. A refractive index is a trick that we play with oil, and this shows the refractive index differences that plays a trick on our eyes is the difference between the water and the air. So the wavelength, or the rays of the sun or the light rays come in, and they're actually bent as they get into the water, and it gives us the appearance that the straw is offset, and that's just due to the difference in the refractive index of air versus the water. Oil does actually us a much better favor. The oil will bend the light inward to the light tube, and we'll get more light. Now, the function of a light microscope, it's all about how much light we get into the body tube of a microscope. So oil does that trick. That plays the trick with the refractive index versus air. Air tends to bend things out, oil will bend them in, so that's to our benefit. And we get about 250x with our scopes using that little oil trick. And that little bit actually was what the original microscope could do in total, was 250x. We use that 250x to pump out that final little bit of resolution. You can see that really makes a huge difference in our ability to view this. And we see this again, if it's a little bit blurry, it looks like there's some sort of, you're going to see this, and this is staph, and with oil, boom, man, it pops, and that's what we want you to see. And again, there's a difference between without and with oil, so please make sure that we use that. We do want to use the iris diaphragm. We can adjust the contrast. It's sort of like the final adjustment for that, which is under the stage. Hopefully you won't need to. But different types of microscopy we're going to talk about outside of light microscopes. We have bright fields, phase contrast. We have this the image conversion type that gives sort of a stenciling look, and we're going to talk about that. We're going to do this is a contrast, still a type of contrast microscopy. There's the dark field and polarized light, and there are other forms which we'll talk about as well. But the bright field, it's mostly widely used is where light is transmitted through the specimen. So that's a sort of a positive, what we refer to as a bright field, and we've already talked about that. A dark field can be adapted from a light field by adding a special disc called a stop to the condenser, and the resulting image is striking. You can see the contrast is illuminated around. So this is the dark field type, and you want to look at internal structures. This is the way to go. It's really nice. It's very useful. Phase contrast type scopes literally take light, and because you can see the notch. Now, if you've gone into a 3D type theater, you have a red lens and a green lens in one eye, and that sort of thing, different color lenses. What you're doing is you're filtering just ever so much of a difference in the frequency of light that comes in, so the phase is just off a little bit, and that's enough now to give you a 3D type effect. And you can see that here is just kind of lets things pop out a little bit, and it's an easy scope to use. And if you need to look at internal structures, that might be the way to go. And this is an epithelial cell. It's kind of hard to see it on a 2D type. If you're actually looking at it under a scope, it actually has a better 3D effect. So again, there's a nice picture of a phase contrast as compared to the two I just was mentioning. And if you have cilia around, the dark field doesn't do it, but the phase contrast will. It's just enough to have those types of organelles pop, and that's what you want. Fluorescence is one of my favorites. Fluorescence, we use a special type of fluorescent type of dye, and what happens is that when you illuminate that with ultraviolet light, it starts to vibrate. And we have different types of fluorescing types of dyes, red, blue, green, various things. And if we do it in contrast to a black field, you can see it just is amazing. So we can have antibodies attached to a particular fluorescent stain or dye, and then apply that, and so the antibody will stick to wherever it needs to, that it is raised against, and carrying with it is a little fluorescein type of fluorescent stain, and then use UV, and boom, you can make some really positive differential diagnosis with that. Here's a simple scope that we used at the vet school. It had the post-processing and various things that we could do, but it's a very nice scope. They tend to be a little expensive. We're $200,000-$300,000, and again, what we're doing is taking advantage of that dye that under UV, just like walking into those types of stores where they have black lights and things just kind of pop out of your clothes in terms of white or whatever. But in a microbiology type lab, we can use it to differentiate between two different types of bacteria, as you can see here, or identify a pathogen using the types of fluorescence tagged antibodies. Another type is a confocal microscope, and we have problems with looking at cell structures that the cells are too thick, and we can use a laser beam of light or scan to various depths of the specimen to deliver. The idea behind it is that we can use fluorescence and also see the structures if they're a little bit more dense. So confocal allows us to do that, and you can see the image. It's amazing. We see the DNA, and this of course is a very powerful tool to use. Now we're going to switch gears and move away from different frequencies of light or UV or various things. We're going to move now to electrons, and now we're going to use an electron gun with various different types of power. The first one we'll talk about scanning. Scanning is not as high powered as transmission, but it's amazing what we can do with it. So the SEM bombards the surface of some specimen. It's been coated with some sort of metal, and so wherever the metal sits, the electrons bounce off and then projects that image onto a screen that's reflective. So you're actually kind of seeing the negative image as bounced off with these electrons. And so you can see this was post-processed. If you ever see scanning electron microscopy output, it's black and white or it's in grayscale. And then now we have post-processing programs that can assign a different color to a grayscale, and gives you that nice color look. And so it's very simple. Your specimens go in the center here. It's usually vacuum, almost a perfect vacuum in a scanning. It's not perfect totally, but it's good enough. And then the visualization part of what bombards the screen. And so we can put a specimen there, usually live or not, and it won't live very long. And then bombard it with the electrons, and you'll see the scanning as it takes the electrons. And of course, wherever those molecules of metal are, it's going to hit, and then wherever the area is that it's going to reflect, you can see that with the image on the screen. So here's we have a mosquito, and if you put an imaginary switch in your mind here, just to toggle a rotations type switch, it's got 3 settings. Let me click it to the next setting. That's all I'm going to do with a scanning electron microscope, is just click it to the next setting, and you can see that I can zoom in. And I have one more setting, so hold on to your hat. Boom. Now I'm really focused in about the best I can do with this. And so you can see I can click, and come back out, and then click, click, go back in. Now I have about, I don't know, 2-3 minutes that I can enjoy doing this before I fry it. But meanwhile, I'm collecting all the photography and I'm getting what I need out of that. So here's the head of a fly. You can see the detail. It's just amazing what we miss because we can't see it. But the electron microscopes, so like the scanning, can pick it up. There's our friend, the deer tick, the Ixodes type that carries Lyme disease. And you can see the proboscis here with the little scalies that allows the proboscis to go in but not out, sort of like a fishing hook. And that's where we get into trouble. And the tricks with using hot matches under bellies and all that, best just to put right across where it's sticking in is some sort of tweezer and clamp that off, because we don't want it regurgitating nastiness into you, and then yank it off and then go see a doctor. Here's Treponema pallidum that was prepared and pushed through a filter onto a filter paper that you could see under the scanning, and you can see this corkscrew type of organism. This one causes syphilis. And of course, under certain types hydrothermal worms, you look at the detail. It's just amazing, the forms of life and what nature has, even at the nanoscale. It's just incredible. Now we'll shift gears again with transmission microscopes. Now, IBM built up to a 5 million electron volt TEM. It didn't really do much better than the 3 million volt, but they had to try. No one really knew. But with just know that we're using brute force to look at. It works in the same way as the scanning, but the transmission blasts a huge amount of the electrons, and it works the same way. The electrons bounce off the sample which we prepare. Now, the big difference between TEM and SEM is the amount of time it takes to prepare the sample. Under TEM, it's quite a process. Now, it was developed in the 30s. The old Ruska electron microscope, the transmission scope, NC State actually had some of those. I worked on one of the originals. You would take a rubber mallet and hit various parts of the stage where the magnets are to align it. It sounds a little crude, but today we just use magnets that we apply voltage to adjust the light, the transmission of the electrons through the path there, and then you can align it and get it through your sample right about here, which is held in a gold grid, and then the image and camera is set up. But this is in a perfect vacuum, and that's one of the other things. It's expensive. You have to put it in ground floor in buildings, so if you ever want to know where they are, just go in the basement. They'll put them on the cement floor and use that there. And here's some pictures. This is a Philips. I used one like this at the vet school, but you can see various different setups. The Joel is a really nice one, but they tend to be about $1 million, and they can go on up to $4 or $5 million, depending on how good of a experience that you want. And I mentioned the preparing of the sample is the hardest part. So you use what they call a microtome, and makes little slices, and you float those slices. And this is sort of a diamond edge, and that cuts your sample ever so thin, about 100 angstroms thick. Float that on water, and if it has sort of a gold color to it, then that's right about the right thickness. And then you take your grid and you pick up your sample underneath that, and then put that on. That's the actual sort of like the stage that we use on a light microscope, but this is for electron, and that little stage fits onto that. It's a little grid that we use for that, and then you have about 30 seconds before your sample gets fried. So be prepared to spend many hours. Did I mention many hours? Many hours. And here you go, though. Look what we can see. You can actually see the binding. This is a virus, and you can see the little attachments even under the scope. There's other types looking at structures, and it's quite amazing. This is really an amazing photograph. We were looking inside a plant cell. I told you that we cut with a diamond knife, and you know if you have a good prep is you don't see any cut marks or striations throughout as a result introducing when you made the cut with the diamond knife. So if there's no imperfections, then that's really nice, and whoever did this, that's beautiful. That is just amazing. Here's the T6 phage. You would grow it and then push it through a filter, and then float that in some type of something that congeals really hard, and then use a diamond knife and cut that, and then float that on a grid, and there you go. You'll see those viruses there. And there's a bacteriophage done the same way. Just make you fix it in a really tough type of material, and then you slice it with a diamond knife, and that's what you'll get. So it's quite amazing. There's rabies virus at 25,000x. It's looks like I don't know what it looks like. Look, bullets or something like that. It's nasty. Now we'll move to the final one that I want to talk about, is the atomic force microscopy type. This was actually developed by IBM about 9 years ago, right here in RTP. And using this type of scope, which costs about $3-4 million at the time. This is the Skunk Works that produced such a wonderful device. And how it works is you have a cantilever that's attached to this little probe, sticky thing that sticks out, and that's about 1 angstrom thick. And you start dragging that over your sample. Now the laser comes down and hits that cantilever, and if it's moving as it goes over the terrain, whatever, it catches that pitch, and then that goes to the detection system and represents the sample that you're trying to look at. And that allows you, the resolution is now that point of that that's attached to the cantilever. So that's usually 1 angstrom thick. So we can look at things at the atomic scale using this, and there's a lot of software and algorithms that pick out the features. So let's look at some of the things that this little tip can do versus the on the surface as we drag it across. And oh, by the way, they're getting more affordable, and we can actually get one if we wrote a grant or something in the STEM Center if we wanted to get a small AFM. So this is what one AFM output looks like, and you can see areas, and what we're supposedly looking at is what the chemists say that we actually see when we use AFM. So they didn't lie to us with the ring structures and representations. It's quite amazing to have that confirmation here all together. So it's quite good. So that wraps up our tour through the microscope. I hope that gives you the various types of microscopes and feel, and how that actually comes out with the data and everything. And so I hope you enjoyed that. And there may be some questions that you can find on Blackboard that will ask you about this video at the end here, just to reinforce your understanding. Thank you. Have a great day.